talk about congees today. Conge is a class of Conantokins, and what that means is that it's isolated from the sea snail family of Conus. So this particular species from which we isolated this peptide was a sea snail, cone sea snail of marine origin. So what this snail does is in nature, sort of lays low among the corals and that sort of thing, and then harpoons a fish and injects hundreds of different chemicals into the fish that is significantly larger than the sea snail and kills it. So the way that it does this is that because those hundreds of peptides, most of them are neurotoxins. Interestingly enough, what we did was we isolated all of these different compounds and found that one of them is actually extremely helpful in preventing neural damage after ischemic insult, which was stroke, or development or continuation of Alzheimer's disease. So, very interesting that something that's used to kill fish ends up being something extraordinarily useful in the medical field. It's a relatively short peptide, it's only 17 amino acids long. Now, if you look at the CD, which I have displayed for you here, now, most of you are probably thinking, what on earth is GLA? Did I skip one? Did I miss one from the 20 that I'm supposed to know off by heart? But don't worry, that's actually rather unusual. GLA stands for carboxyglutamic acid. And there's 5 of them, these residues, within one peptide, or a conjugate peptide. And this is the reason why a lot of labs can't afford to use this, or to research this compound, because GLA is exclusively synthesized within the metabolic systems of the cone snail or synthetically, and it's not cheap. Because of these 5 residues, 1 milligram of, 1 milligram of conge is $350. And the working concentration, in my experience with this compound, is 40 micromoles per liter. So, if you're looking at concentrations like this and you have to use it with every single plate, it's going to get extremely expensive. So there's only 2 or 3 labs in the world that work with this particular compound. And they've had some fantastic phenomenal results with it, but GLA is what makes this prohibitively expensive. There are other variations of Conantokins. I think there are Con-G, Con-T, Con-R, and Con-L. What's really interesting about Con-G is it selectively binds the NR2B subunit of an NMDA receptor, N-methyl D-aspartate receptor. I'll get into those later. But that's what makes this the superstar of the Conantokins, because of its selectivity and the fact that it affects one of the ion channels that are implicated in Alzheimer's and ischemic insult, or rather neural death following ischemic insult. So this is them. This is the N-methyl D-aspartate receptors, which I'll call NMDA, for just to save my breath. These are interesting receptors for multiple reasons. One, it's not a single ion channel. For example, you can have, it's non-specific. You have calcium, sodium, and potassium traveling through this channel. A lot of sodium channels that you find in the brain, or not sodium channels, but ion channels within the brain, are selective for a certain ion. They will be calcium channels, or sodium, or potassium. This is unusual in that it is non-specific. So you can have sodium, calcium, and potassium traveling through the same ion channel. The other aspect that's interesting is that this is both ligand and voltage gated. A lot of channels, you get one or the other. Ligand meaning that it has to bind certain molecules for it to open. Voltage meaning that it has to have a certain action potential across the membrane in order to have activation of the channel. This is very interesting because it's got both. And I personally think the reason why it had evolved this way is because it is a non-specific ion channel, it has to be under tighter control. And that's why I think this probably evolved to have 2 methods of control as opposed to 1. So, why the voltage-gated aspect of control of this receptor? Because you have a magnesium bridge here, Mg2 site. This needs to be removed, evidently, before you can have something go through that. And the way that you remove this is a certain action potential, a certain voltage limit has to be achieved across the membrane in order for that to be removed. That's the voltage-gated aspect. On top of that, it has to, the bare minimum ligand portion, or what you have to have bound for activity, is glutamate and glycine. You can see in this image here, there's also a polyamine site and a zinc site, but what I was adding in some of my experiments, as long as it had glutamate and glycine, those were the minimal ligand requirements to open this channel. The reason it's called NMDA is because N-methyl D-aspartate mimics glutamate and is able to fill that same site for activation of this channel. Mention that. The structure of your NMDA receptors is that they're tetramer proteins, and you have two pairs. You have two obligatory NR1 subunits on one side. So I realize this is difficult to see, but really this is a tetramer protein. Two obligatory identical NR1 subunits. And if you look at the mRNA coding for these subunits, there's 8 different splice variants. So what that means is if anyone built a gingerbread house over the holidays, I know sometimes what they'll do is they'll give you a set of different bricks of gingerbread, and then they'll give you 3 or 4 models that you can build with those pieces. So you can have 3 or 4 different looking houses from the same kit. This is very similar. From the same mRNA strand, or sequence rather, after post-transcriptional processing, you can have 8 different splice variants of the same NR1 subunit. And these are found in different parts of the brain. The aspect that I was looking at, or the type of neurons that I was looking at, was from the hippocampal region. So I'm so glad that Danny did this presentation before I did, because now it makes sense. So that's one aspect to keep in mind. Now the other 2, so I've explained the 2 out of 4, so what's going on with the other 4? They're the NR2 subunits, of which there are 4 types, A, B, C, and D. A and B are by far the most prevalent in the brain and central nervous system. C and D, recently discovered, rather remote. So in comparison with the NR1 subunits, which you have to have 2 copies of the same one in that tetramer protein, so 2 of them are going to be NR1. For the NR2 components, they can either be 2A, 2B, or A and B. And this differentiation is seen whether it's extrasynaptic or synaptically activated, which I'll explain in a moment. So this differentiation happens not only in different parts of the brain, but in different parts of the neuron. Okay, so now the actual research part. Phosphorylated ERK. So what I was explaining about extrasynaptic and synaptic activation, in those lovely diagrams that Danny showed us, he showed you what the synapse was, which is essentially, it looks like, if you will, a tree. So you have the bushy leafy part, and then you have the trunk, which is the axon, and then you have millions of branches, which are similar to your dendrites. I'm not sure how well you can see this picture, but here is an example of the somal body. So this is the body of the cell, and all these little, they're not particularly well shown up here, but branches are dendrites. See that big thick one coming off of that neuron? That's the axon. Usually there's only one of them, and they're very, very thick in comparison to your dendrites, and that's how you can see whether you're looking at a dendrite or an axon. So what ends up happening is that at the synapse, so the definition of synapse is where you have the connection of one neuron to another. That's the definition, working definition of synapse. You find a series of NMDA receptors, but you also find them along the dendrite where there is no synapse, which is called the extrasynaptic site. So you have your synapse, which looks like this sort of model. This is the one dendrite, this is the synapse, and there are neurotransmitters that are being sent between these 2 signals. But then there's a whole bunch of different dendrites along there which don't have contact with another neuron. Those are called the extrasynaptic sites. And chemically, when you isolate these neurons from rat pups, let's say, you can chemically selectively activate just the synaptic NMDRs or just the extrasynaptic ones, which has been extremely helpful because physiologically, when you have neural death as a result of Alzheimer's disease or ischemic stroke, something that's causing brain cells to die, it mimics the extrasynaptic situation, i.e. anything that gives a global glutamate release in the body and causes these NMDA receptors, because if you recall, one of the sites was glutamate, one of the ligands necessary was glutamate. You have a global glutamate release in the brain after a situation like a stroke or Alzheimer's disease, which activates just the extrasynaptic NMDA receptors. So this has been very advantageous to do this chemically because it mimics in tissue culture what you're seeing physiologically on a very much less complicated scale.